Let me start by talking a little bit about this artifact that's on stage. Sometimes we bring some things out of our vast collection, 120,000 items related to the history of computing. This is the Paracel Gene Matcher. It's part of our permanent collection. It began as a top secret government project at TRW. At that time it was called the FDF, the Fast Data Finder. It was introduced in 1997 and it was based on the ASIC circuit technology, if you're into circuit technology, and it was specifically designed to analyze genetic information, similarities and differences of DNA and protein sequences in relation to other sequences and other known genetic functions. It was originally used by the University of Washington's Institute of Systems Biology for the Human Genome Project, and we'll talk a little bit about the Human Genome Project tonight. As you can see, it's not very big. If it had some wheels, maybe you could roll it on to the plane you're taking and put it in the overhead compartment. And it contained about 27,000 programmable parallel processors. So that is the Paracel Gene Matcher, and it's making a guest appearance tonight because we're talking about this subject. It is the vital intersection of medicine and technology, and of course tonight we're focusing on genetics and cancer. Scientists and medical doctors have known for a long time now that cancer is a genetic disease, that its cause is related to the way genes control the way our cells function, and especially how they grow and divide. All cells grow and divide, of course, because of genetics, but cancer cells have many more changes than normal, and each person's cancer is unique in its combination of genetic alterations. So understanding these genetic links has become central to a search for better treatment and better understanding of the disease itself. The Cancer Genome Atlas, which we'll explore tonight, is one example of that work. Investigators with the Atlas's research network have conducted large-scale genomic studies of more than 30 types of cancer, including 10 rare cancers. They've also published a cross-tumor analysis of 12 types of cancer. Collectively, this effort has produced what we now know today as big data. Big data about the relationship between cancer and genetics, and more than 1,000 papers have been published from this data set, and the work goes on. With us tonight to help explain the importance of this intersection of computing, genetics, and cancer is Dr. Jean-Claude Zenkluzen. Dr. Zenkluzen has worked in the field of genetics and cancer since 1996, when he participated in the Human Genome Project and cloned 2 novel cancer-suppressing genes. Since 2013, he's been the director of the Cancer Genome Atlas, which, as I said, is the largest scale cancer genomics project to date, involving hundreds of investigators from more than 30 countries. We're so pleased to have him with us tonight, and this room is a lot cooler than his home base in Washington, D.C., so he's especially glad for that. Please join me in welcoming Dr. Jean-Claude Zenkluzen.
Thank you, John.
Welcome. Welcome to the museum.
Yeah, and as you said, I'm very happy to be here in California. DC these days is a swamp. It's very, very, very humid.
Some would say it's a swamp in more ways than one, but
we're not going to go there.
Let's not get into that.
As a federal employee, I cannot comment.
All right, I can say that, but you can't. Did you ever work on one of these, by the way?
No, actually, I was looking at it. We never used this one, and I think the reason is because, as you said, the company was acquired by Celera. And when we were doing the genome, we were, the public side and Celera, we were competing with each other, so we were using different systems. But yeah, I never had the joy of using one of those. We were using old Unix boxes, which right now they are quaint, but they were very useful at the time.
Yeah. We're going to talk a little bit. One of the things we like to do when we have an evening like this is get to know you a little bit personally, get to know your background. So can you talk a bit about where you grew up, Patagonia, your education, and how you've made your way to this point, maybe leading up to the point where you started getting into genomics?
Every single time I would talk about how my journey to the chair of TCGA, it looks like everything was done on purpose. It's like totally serendipitous. So I'm Swiss originally. My father was hired to build a ski resort in Patagonia. So 3 years old, here we go. Patagonia, middle of nowhere. I loved it because it was a very small town, in front of a beautiful lake, surrounded by mountains. So all the kids were free-roamed kids, free range. You know, we were going around town. Everybody knew each other. If your mother needed you, opened the window, shouted your name, and your name got repeated until somebody found you, and then they brought you back home. So it was great growing up there. Very similar to Switzerland, obviously. And then at some point, my father finished the ski resort, and we moved to Buenos Aires, where I did most of my school. I did my high school in Buenos Aires, and I also did my undergrad studies in the School of Sciences for the University of Buenos Aires. I'm a chemist originally, hardcore chemist.
Hardcore, what is hardcore?
Hardcore protein chemistry, very chemist. No applied nothing. I imagine some of you know the Big Bang Theory series. You know, I would have been the Sheldon of the chemistry world. It's like I was doing very basic chemistry science, and I was interested in neurobiology because I do believe that the brain makes us different from the rest of the animal world. So and we don't understand really how the brain works, and so I was interested in doing that. And then something happened in my life that changed my vision. Cousin of my mother, Susanna, she came down with a very bad sarcoma. She was closer to me in age. I was 20, she was 28. And she was also a chemist. She had just finished her chemistry degree. I had inherited all her books. And so she comes down with this horrendous disease. And since her family lived in a province in Buenos Aires that did not have as good treatments as they were in Buenos Aires, she stayed with us at home. And so I saw this beautiful, vibrant lady go from a great, nice, vital person 2 years to a small skeleton of a person that basically, she looked like somebody that came out of Auschwitz. Because of the disease and because of the treatment. And so when she finally died, I was close to finishing my master's in chemistry, and I decided that neurobiology is all nice, but now I have a personal enemy. And so that's exactly why I decided to go into cancer research.
So you took this very personally.
I still do. This is something that, you know, some people say, oh, I have the Monday morning problem or the Sunday afternoon blues because you have to go to work. I never had that. I really go to work fully aware of what we are doing and fully aware of the necessity of what we are doing. When we were studying, my wife is also a scientist, also a chemist. When we were studying at MD Anderson, I was doing genetics, she was doing biology, she was working with mouse models. And mouse can be disappointing. They not always do what do you tell them to do. And so sometimes she will go, oh, I don't know why we're doing that. And I will go, well, let's go to the hospital, sit in the lobby. And she goes, no, not the hospital again. But yes, the hospital again, because that's the reason you do it. I never had that. And sometimes I tell you, you know, Washington having to deal with budgets and stuff like that sometimes is not too happy. But I have a picture of Susanna there in my desk, and whenever somebody really bugs me, and I wonder why am I doing that, I turn around, I look at the picture, and I go, yeah, I know why I'm doing that. And I think most people that are doing this type of research have a personal connection to it, because cancer is a very common disease. 1 in 3, 1 in 4 people are going to come down with cancer, not die from cancer, but they will suffer from it.
It's interesting. My mother was treated at MD Anderson, so that's a personal connection for me, absolutely. Well, that's wonderful because we like the stories of the human connections behind this kind of science and technology. Let me ask you a little bit about, you went to the National Genome Research Institute and got your start in the Human Genome Project after you finished your PhD. What was happening at that project then?
This is very early days in the Genome Project. In fact, the institute was not an institute, it was a center, because this is the first step in the NIH. You become a center, and then if you do good work, you may grow up to an institute. And so we were a center. We had one project, which was doing the human genome. I went to work with Eric Green, who right now is the director of the Genome Institute. And at that time, Francis Collins, who now is the NIH director, was the director of the institute. The institute was tiny, 400 people in total. And we had one role, which was sequence the genome. And so what I was doing was putting together a map in chromosome 7 with everybody else in the Green's lab. Why chromosome 7? Because you said, I cloned 2 tumor suppressor genes, which happened to be in 7, so I already knew the area. And the cloning happened while at the Center for Human Genome, because we didn't have the sequence. In fact, the piece of DNA that I mapped in Eric's lab was the first piece ever to be completely sequenced. And the reason was because I already had a good part of the map when I came to Eric's lab, so I had a leg up, basically. I could put together the map together. It's interesting because we were just talking about Celera and the
Yes.
public side. So the public project, the idea was we were going to put together a map of the whole genome, large pieces of DNA in what we don't use almost anymore. They were called BACs and YACs, the bacterial artificial chromosomes and yeast artificial chromosomes, that basically we made a chromosome that bacteria will carry and reproduce that contain a large piece of human DNA. And then we mapped markers into it, and then we did a tiling of minimal overlap. So you knew each piece of DNA where it belonged in the chromosome. And then what you did is you went and sequenced. And so that is a very tedious way to do a genome. It's time-consuming, very, very, very work-intensive.
And how were you doing it? What was the tool that you used?
So what we basically were doing it with using markers, very particular sequences of DNA that were distributed around the DNA, and that was possible because, as Newton said, I saw this far because I was standing on the shoulders of giants. This is how science really goes. A group in France, the CEF, had created 400,000 markers across the genome that they knew in which chromosome and in which order they were to each other. They didn't know exactly where in the chromosome, but they knew the order. And so we used that collection to look at these pieces of DNA and do a PCR, an amplification of the segment, to see if each one of those packs had that marker or not. And so if they had the marker, we put them in the bin of contains the marker. And then as we kept going, we could tell how much of an overlap there were between the different bags. The bags were totally random. Basically, we took human DNA, chopped it to pieces, put the sequence that the bacteria would recognize as its own, and then put that into bacteria to replicate and have enough of it. So it was quite a work. Celera had the complementary approach. They said, well, you don't need to know where the DNA is. If you sequence enough DNA, you're going to be able to attach the different pieces together, and ta-da, you're going to get, your chromosome.
So it's a computational problem.
It's a computational problem. And they did that. They basically developed what we use today to sequence, which is the shotgun sequencing. And it's as it sounds. You take DNA, You load it in a shotgun? No, you don't do that. But you take DNA, you break it into small pieces, and then these small pieces, you create a library and you sequence. And it's a very small fragment. It's about, right now we sequence about 30 to 70 base pairs in one shot. And so you get a lot of very small sequences. But if the sequences are unique, you can overlap them. And again, you create what we call a contig, a continuous set of DNA. That's all fine and dandy as long as the sequences are unique. The problem is that all genomes have a lot of repetitive sequences. And so you are unique, 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 unique. You hit the repetitive sequences, and now you don't know where to go because everything looks the same. And so the two approaches were going parallel. This one, the mapping that we were doing, we were going to end up having the genome, but it was going to take a long, long time. This one, the shotgun, was not going to end up having the genome. So after a year discussion between Celera and the public projects, we agreed to come to a truce and use each other's data. And so that's where the genome was built really, really fast because we knew the scaffolding and they knew the content. And so we put each other work together and we got the first draft.
And what year was that?
That was 2003.
2003. So
it's fairly recent.
In scientific terms, this is very recent.
Oh yeah, it's a breath. It's just one breathing. We basically have, the genome has had it for 13 years only.
So let's move from mapping the genome, which now is complete, to the relationship between cancer and genetics. And you've given us a slide that I'd like to bring up now. You'll be able to see it on all three screens that shows a little bit of the relationship between DNA, the cancer-causing agent, what a normal cell looks like in a cancer cell. This is obviously very stylized. But could you walk us through this and also work into this how you moved from mapping to the relationship with cancer? So what happens is, in order for cancer to, let's step back one step. Cancer is a disease of genes. Genes get mutated. But also cancer is mostly a disease of older age. I'm not going to say old age because I'm getting there. But you need to have lived for a amount of time in order to be at risk for cancer. And some people will say, well, but you have childhood cancers. True, but childhood cancers are rare cancers. They're truly very rare. And they mostly happen in blood. So they are hematological malignancies. Because blood cells are easier to dysregulate than organ cells. And so the reason most cancers happen after 50, 60 is because you need to accumulate a series of events that are mutating different genes in the same cell. If you have them in different cells, there's no problem because the effects are not felt. So all these events have to happen in a single cell. And this is what is shown here, basically. You have DNA, and here we are looking only at one of the forms of the genes that get altered, a proto-oncogene, which is a normal gene. It's a gene that the cell needs to divide. It's a gene that is absolutely necessary. And then through events, exposure to sunlight, smoking, exposure to carcinogens at the workplace, or just plain bad luck, because mutations happen randomly, you get a mutation that transforms that regular proto-oncogene into an oncogene. An oncogene is a gene that pushes the cell to become cancer. And so what happens is if you get enough of these events happening in a single cell, you can then have this cell transform into a cancer cell. And that's one of the types of the genes. The other type of genes, as you alluded, it's the tumor suppressor genes. They are genes that, as long as they are active, they will block the formation of tumors. And so these ones, you need to eliminate the gene. And so it's a combination between activating genes that normally help division and eliminating genes that control the way the cell behaves that you may end up having a cancer cell.
Still, that does not mean that you have cancer. You may have a tumor. And by the time we die, we carry about 10 to 15 tumors, which they're not going to kill us. You all probably have or know of skin tags. Well, those are tumors. It's a growth of cells outside the compartment. But that's not going to kill you, you know. Many of those are virally induced. We are learning, thanks to TCGA, that we have a lot more virus involved in cancer than we thought. But what happens then is these cancer cells are, when they become cancerous, they are very specific to the person that carries it, because the sequence of events that happen for that cell to become a cancerous cell is very specific to that person in particular. And so we used to classify tumors as breast tumors, prostate, renal. And then through the Cancer Genome Atlas, we have learned that there is no such an animal. Breast cancer, for example, is 5 different diseases, totally different from each other, totally different biology, and they should be treated differently. And we kind of have a hint of that, because you have people that respond to therapy, people that don't, and so we kind of knew. But now we know what are the alterations that are defining those groups. And we do that alteration analysis through sequencing of DNA, through sequencing of RNA, through analysis of copy number changes, which is you have more of a certain part of the DNA or less of a certain part of the DNA. And so through that analysis, we can tell what biological hallmarks have been altered in that particular tumor type.
We've skipped ahead. We're looking into the Genome Atlas, and I want to get there. So let's do that. But first, tell us how it originated and how you became attached to the project.
That's an interesting thing. Basically, when the genome was available, now we have all the complements of genes. Some technologies, which looking back now, 10 years, they look like absolute primitive things, like writing with a stone tablet.
So you see that now, you look back at it?
Yeah, I look back at it and I go,
where were we?
How did we do that?
Yeah. But at the time, it must have
But
been
at the time, it was amazing.
Yeah.
You had the whole complement of genes. We mostly use systems called microarrays, which were glass plates that had different probes laid onto them, and then we hybridized the RNA onto it or the DNA onto it.
Very manual process.
Very manual process. Then some companies came up. Affymetrix was one of the most successful ones, still around, that came with things that were a little less manual, but it required a lot of manual input. And so we got data on what parts of the DNA were missing. We got data on what the different levels of expression of RNA were, and so a lot of people started using these tools to try to investigate different tumor types. I told you that I was interested in neurobiology, and I must have done something good in life, because I started working on cancer, and then I got offered a position to be the chief of genomics at the neuro-oncology branch at NIH with Howard Fine. And so suddenly I'm studying tumors, but of the brain. And so I just combined two things that I was really interested in. And so we did a project. Howard had a good idea of asking for the funds and asking for the samples. We did a project to analyze 1,000 gliomas. Gliomas come in 2 flavors, basically. You have the low-grade gliomas, which have a 10 to 15-year development time. And then you have glioblastoma multiformes, GBMs, which are lethal. You probably know some people with GBMs. Edward Kennedy, Senator Edward Kennedy, died from a GBM. Bo Biden died from a GBM. It's a devastating disease. You get diagnostic, and normally a year to a year and a half later, you're done. You're dead. It's really rapidly advancing. It's not very common, but it's devastating. So we did the analysis of this, and we created a database called Rembrandt that had all the data where people could go and analyze it. And it became quite a success because it was very easy to use. And so NIH decided that it would be a good idea, NCI decided that it would be a good idea to do this in other tumor types, not just in GBMs. In fact, one of the questions that I get all the time is why TCGA started with GBMs, which are not a common tumor. And my answer is because we had 1,000 of them in the freezer already. And so, it was an easy place to start. So the project started. I was not part of the project at that point. And then some years later, they offered me to work at the Office of Cancer Genomics in some genomic projects. I thought it was a good idea. I moved from the lab to the scientific management side. And sure enough, a couple of years later, they offered me to go back to TCGA.
And the goal of the Atlas was what?
The goal of the Atlas was to characterize, initially, it had 3 stages. The pilot phase was to demonstrate that you could do these projects. And so ovarian, GBM, and lung adenocarcinoma were part of the pilot, and the idea was a demonstration project. This can be done. This can be done at scale. This can be done in a way that the data can be analyzed. Very interesting thing in the pilot phase, we learned a lot of stuff that we never thought about. When the first project went ahead, the prevailing notion of cancer was we're going to find 4 or 5 genes that they will be mutating at 30–40% of the patients, and that will explain the biology. Well, we do GBM, we do the sequence, we don't find that. We find a couple of genes that have a 30% incidence, and we find tons of genes that have 3, 2, 5% incidence. And so when we got the data and we started looking at it, humans that we are, we thought, oh, okay, the data is wrong. So went back, looked at the data to see if it was technically correct, and it was technically
Why was this
correct.
so surprising?
It was very surprising because to that moment, there were just a couple of genes that were the recurring cast of characters that were appearing in different cancer types. And so the other ones had not been discovered. And so the idea was, okay, we have maybe 100 oncogenes. Well, we have a lot more, and they are very specific to the different tumor types. But one of the things that we discovered is that although you have all these genes with low levels of alterations, when you start looking at the functions that they fulfill, and I say functions and not pathways because in biology, pathway is a word phrase with conflict. But basically, you have a function that has to be fulfilled, like maintaining DNA integrity. And then if you started putting the genes in the different pockets for functions, they made sense. So you had a lot of 2, 3, 5%ers, but we're all altering the same mechanism. And so something that for us now is obvious, cancer is not a gene alteration, but it's a mechanism alteration disease. At that time, it was not so obvious, and we were extremely surprised to find that.
And was this the result of simply being able to aggregate all this data and slice and dice and compare?
That is exactly it. It was the result of having a lot of samples, a lot of data, and also data at the depth of coverage that we had not had before.
And how had the change in computational power available to you as a scientist made a difference?
It made an enormous difference. To give you an example, the first TCGA project, the GBM project, took 2.5 years to do the analysis of the data. software algorithms were not up to snuff. Computers were not as fast as they are today. It took a long while. In the past 2 years, we have published 17 different papers, which are 17 different tumor types. So in the past 2 years, we have done 17 tumor types analysis, one after the other. It takes us about 2 months.
And in the beginning, it took you 2.5 years to do a single
exactly.
study.
And one thing that I repeat myself all the time is that the difference in what cost and time was when we did the first genome to what we are doing now, it's amazing. The first genome took about 10 years, and it cost about $300 million. Nowadays, we get a genome in 2 days, and it costs about $1,000. So technology has advanced enormously. But the chemistry is the same. So what it has changed is how the computers are able to read the reactions. We are a lot faster on reading the reactions. We are a lot faster on accumulating the data, and we are a lot faster on doing the analysis of the data. And that is all due to computational power. I cannot code to save my life, but I have a lot of respect for people that do, because they make my life so much easier.
Now, you were telling me earlier that the tools that are actually used for this computation have actually been around for a while. They were developed in software terms a long time ago.
Some of them, yeah. For example, trying to figure out in a piece of DNA if you have a gene and what the gene model is. This is old. This was developed even before the genome was finished. And as I was telling you, the reason is because the rules of what makes a gene, where it starts, where it splices out an intron, where it finishes, have been known for a long time. I'll say since the 70s. And so the algorithms that were developed originally, and the one that is still I remember the other day, I was thinking back, and what did I use? It's Grail. Grail, like the Holy Grail. That algorithm is still around. In fact, when people assemble genomes and try to figure out what the genes are, they still use Grail, because it's an algorithm that was developed in the early 90s, and it's still around because the rules have not changed. There are other algorithms, like predicting splice forms of a gene, basically what forms the RNA will take once it's translated, transcribed, sorry, that we are still evolving. So versions of algorithms for prediction of alternative splicing that are 6 months old, they're prehistoric old, because we are learning more and more through these projects what the different rules of the game are. And so as we learn more rules, the algorithms adapt to it. At some point, hopefully, we will know all the rules, and then we will have the algorithm that does it. It's very interesting, because there are certain things like copy number changes. When I was doing the glioblastoma project that I told you, that was the holy wars were on what was the right way to predict copy number changes. That has been settled. Everybody agrees there's one way to do it, everybody does it. But then we moved on to the other problem, and the other problem, and the other problem. And when we think we are getting close to get all the solutions, we realize that we know nothing of biology, and a new entity comes around, like microRNAs or microRNA sponges, and there are things that we go, oh, I thought I had an idea of what the universe looks like, and I know nothing.
Has this process also taken what was highly manual and made it highly automated?
Absolutely.
Is it mostly automated now?
It's highly automated. You still need the human here and there. You need to know
I find that reassuring.
Yeah, which is good to know. But I will say the good news is we are always going to need the human, because we can do something, or in the foreseeable future, we're going to need the human. Because we can do something that machines cannot do. We can have imagination. If we find something that doesn't fit the model, we don't choke. We try to figure out why it doesn't fit the model. The algorithms, they just stop working. And so you need the human in the middle to do the analysis. But what has happened is that the processes by which we produce the data have become more and more automated to the point that now the reactions are set up by robots, which is good because all the menial tasks of pipetting, God, have I done that? You really don't need to do it in that scale. And we would not be able to produce the data the way we are producing it if we were still relying on manual pipetting. And so it has freed the human from the menial task and allowed us to use our time and our effort for the more creative part, which is the part that the machines cannot do yet.
So what are all those postdocs doing instead of pipetting?
They're mostly looking at the data that is coming out of the computer.
Which you'd want them to do.
Yeah, which is exactly what you want to do.
I want to talk a little bit about how this scaled, because you talked about how it got started. So this was an extremely distributed process, 150 researchers, not simply here, but around the world. How did that...
And that is counting only the laboratory PIs. If you count everybody that worked in the project, we're talking tens of thousands of people.
How did this stay coordinated? How did you share data? Can you talk a bit about that process?
Yeah. Well, at the beginning was very chaotic because the pilot phase was supposed to figure out how to do this. And so it was very ad hoc. We basically had a secure FTP bucket where all the data entered, and then the analysts took the data that we're interested in and started doing the analysis. But as we progressed, what happened was different players said, well, but all those steps that an analyst is doing, we can automate them into a pipeline. And so pipelines started to get created to take the data and give you the initial display features. And we still do it that way. The way we run these projects, and we run all of these projects the same way, is we have entities that provide us with the samples, and these are also the experts in the disease. And so they will be called onto analysis later on. But they provide us the samples.
And they tell you what's interesting about that sample, why they...
They don't tell us anything because we don't want to know anything at this point. Okay? We want to be totally blinded. We know only that it's a certain tumor type, lobular breast carcinoma. Okay? So we know that. It goes to an entity that we call the biospecimen core resource, which they look at pathology. They make sure that it's breast. You would be surprised. We got 19,000 samples submitted. We qualified 12,500 and some. Many of the ones that were not qualified were because they were the wrong organ altogether. Okay? It happens, pathology labs have a lot of samples. They take the wrong tube, then we cut the specimen and we go, this is not kidney. And so we put it aside and forget it. So, that's one reason. The other reason is because the quality of the sample was not good and we didn't get DNA or RNA that was good enough. And so that also happened. And so this core checks first that, the sample is what's supposed to be. And then they extract the DNA and the RNA and they do quality control to make sure that it's good. Once that happens, they get shipped to genomic characterization centers, basically sequencing centers. Large parallelized systems with a lot of robots that are doing all the reactions. They're creating the data. The data gets deposited in a large secure FTP where all the data ends up being. And then we have a series of analysis centers, the genomic data analysis centers, that specialize in different facets of the data. So we have someone that will be specialized in looking at point mutations in DNA. Somebody will look at microRNA patterns. Somebody else will look at messenger RNA expression levels. Somebody will look at copy number changes. So they look at all the same data, but they look at it from different angles. And they produce display items with the data that is created. And at that point, we convene what we call an analysis working group. And the analysis working group is made up of the people that did extractions, the people that generated the data, all the analysts that analyzed the data, and, as importantly, all the clinicians that provided the samples and understand what the samples are. And so now we are looking at the data all together, and this is the time where we want the clinicians to tell us, no, but that subgroup that you have here has this particularity, and so that's why it's looking different. But we don't want to know that at the beginning. When we create the data, we want to be totally blind, and when we do the first pass analysis, we want to be totally blind. Right now, this is a very well-honed pipeline. But it took a lot of errors in the process and knowing how not to do stuff in order to get something that works. Sincerely, at the moment from the time we identify the samples until the time the data is finalized, we have very little to do. It just works in an automated fashion. It's a factory kind of.
And then you mentioned robots manually doing work in labs. Are you starting to use software bots that can go into this data and do the analysis independently of other kinds of human intervention?
This is something fairly new. But we have one of those genomic data analysis centers that I told you about. We call it the processing center. It's run by the Broad. They have a system called FireCloud that basically will automatically, without human intervention, go check the directories, check for the data, and do the processing and issue out the standard analysis that doesn't require a human in the middle. And so we then take that one, those displays, and do the human intervention.
And if it finds something new and unique, does it flag that up and say to you, this is something we've seen?
Sometimes it does. Sometimes it can tell us this is something we have not seen before. Sometimes what it does, it just tells us, I don't know what to do with this. And so, fine.
Someone steps in.
That's when somebody steps in and looks at it and they go, okay, now I understand what happened here.
We're starting to collect the question cards, so if you do have questions, please hold those up. And if you're live streaming on Facebook, you can send the questions in by Facebook now. We'll make sure that those get into the program. I want to talk a bit about outcomes. And there's a slide that we have here that is part of an infographic that the NIH has issued. So as you can see, if you're kind of a data nerd, like many of us here are, you can see that the Atlas now contains 2.5 petabytes of data on cancer and genomics. That's 212,000 DVDs, a lot of DVDs. You can see 33 different tumor types, 10 rare cancers, based on data from 11,000 patients using 7 different data types. How phenomenal is this to have this, and has there ever been anything like this before?
There has never been a biomedical project like this before. This is the largest biomedical project ever. I imagine that 10 years from now we're going to say, well, TCGA was quaint, but we have done this humongous thing because we tend to go bigger and bigger.
Sure.
But so far, this is the biggest project. It's interesting that we have accumulated 2.5 petabytes of data, and this is one of the things that I think is one of our debits to the scientific community and to the community in general, which is, so far, in order to analyze the data, you have to actually download it.
Oh,
yeah. So 2.5 petabytes, no, I cannot put that in my home computer.
Yeah.
Okay, so it's quite difficult. And in fact, large institutions are only the ones that can do it. We're working on solving that solution because that problem. Because when I entered TCGA, I told you we had put together a database that was very easy to use and is still popular, although the data may be a little dated, because it's easy to use. And so when I entered, I said, well, we need to make the data usable. Available and usable, they're two different things.
Sure.
It's available. It's not really usable for small entities or for the public in general. After all, this is paid with taxpayers' money. This is the taxpayers' data. They should be able to go and look at it. And so what we are creating with our new database, the Genomics Data Commons, is the first thing we did is we took all the data which was generated in different platforms and was analyzed against different versions of the genome, and we made it all look the same. So now you can look across all tumor types, and it all looks the same. But what we are going to about to open up, June 26, I think, but end of June.
That's pretty quick.
That's pretty quick. So the Genomic Data Commons opened in June 6 last year. A year on, we have a second version that will have a lot of visualization tools. So now what we are going to be able to do is go in, don't download anything, select the type of tumors that you want to do, select the characteristics of the patients that you want to do, and start analyzing the data in situ, in the systems. And so you're going to be able to see survival curves. We are going to be able to see mutation patterns. So what we want to do is this data to allow the scientific community to come up with hypotheses that then they can test in the lab in a one-on-one basis that cannot be done in a pipeline fashion. But that's what is going to bring this data and this discovery to the patient. Because until we verify this in an organism or in a cell, we really don't know if what we are seeing is relevant for therapy. And so that step requires a lot of individual labs around the world looking at the data and analyzing it. And the only way they can do it is if they don't have to download 2.5 petabytes.
In the era of the cloud and all this shared information and networking, why the download requirement?
It's just because the tools were not co-located with the data.
Ah, okay.
And so this is, the GDC is basically a cloud. It's not basically a cloud, it's a cloud.
Yeah.
It's just not in a commercial cloud. It's a cloud that we manage, it's a private cloud. But the tools were not co-located in the same space.
Okay.
And what we have done in this past year was to co-locate the tools so you can do the analysis. In fact, NCI has had a project for the past 3 years called the NCI Cloud Pilots, which is trying to do exactly that, is put the data together with the tools and allow people to do the analysis. And through what we have learned on the Cloud Pilots is how we developed the Genomics Data Commons.
Is this done now, and now that it's evolved into this Commons project, or is more data being added to this atlas?
More data, well, not to the atlas. The atlas, it's done.
It's finished.
We did all the data that we could do. In fact, the atlas, one of the things that happened was initially was significantly smaller, and we could grow it because of the American Recovery and Reinvestment Act, ARA. They had to be shovel-ready projects. Ta-da, shovel-ready. And so we got money there, and we could expand the number of cancers that we could do. It was a big infusion through that. But we have done the atlas. We're not continuing on the atlas. What we are continuing is on genomics. One of the problems that the atlas had was that the clinical information that accompanied the samples was sparse. And so we could not ask too many clinical questions that could be taken to the patient. And so what we are doing now is we're doing more of these type of projects, but now we are using samples that are coming from clinical trials. We're coming from clinical trials that are either finished or ongoing. And so now we have a rich molecular data accompanied with a rich clinical data, and we can start asking questions. For example, one of the projects that we are running right now, it's in lung and in colon, early stages. And what happens in lung and colon, both of them, in lung adenocarcinoma and colon adenocarcinoma, is that everybody, all the patients, early detection, they get treated the same way. Normally for colon is surgery, the tumor is removed with a surgical margin, and then you get radiation, you get chemotherapy. 40% of the patients are cured, never to see that tumor again. perfectly fine, live the rest of their life until, I don't know, they get run over by a bus, okay? But cancer is not a concern anymore. 60% of them will have a recurrence and will probably die from the disease. But when you look at them under the microscope, they look the same. There must be a biological difference, but we don't know what it is. And so here enters genomics. We have cohorts of patients that we know who recurred, who didn't. We analyze, compare, contrast. And hopefully, I'll say in 2-3 years, I can come and tell you the people that do recur for this. And that is not only important for the treatment of the patient that will recur, but also if we can identify who has not a problem anymore, that person's quality of life, it's a lot better. We don't have to have a colonoscopy every 6 months. I had my first one a couple of years ago. I'm not looking to repeat the experience. So, you know, it is significant to be able to tell somebody, or also prostate cancer. There's some prostate cancer that are indolent. They'll never go anywhere. We don't know which ones they are. But if we can, then we can tell, okay, so you have prostate cancer, but you know what? You're going to be 120 before it kills you. So worry not. Worry about other things. You know, maybe your diabetes, the lack of exercise, or whatever it is that you're not doing. And so all of that, those questions we could not answer with the Atlas, but all these new programs are going to allow us to do that because we have good clinical data.
What are some of the breakthroughs that have already occurred as a result of this?
I tell you, when the Atlas was started, I did not think that we were going to find things that could be applicable to the clinic immediately.
Why? Really, that's interesting.
Because when it started 10 years ago, there were very few targeted drugs, and so we didn't have the tools.
Yeah.
We still don't. We have the tools for some of the alterations, but not for all of them.
And when you say targeted, what do you mean by targeting?
I mean a drug that specifically works on that mutation that is causing the tumor. So we didn't have that. And also I knew, because originally when the project was authorized, we were authorized for 3 clinical data points: age, disease, and gender. With that, we're not going to discover anything that will make any difference in the clinic. It happened that we actually collected a lot more than that. There are some things that are really, really important and have translated into clinic immediately, really, really fast. One of them happened in low-grade gliomas. When the classification was finished and published, there had been hints that some low-grade glioma, they're not the 10, 15 years development that I told you, they evolved a lot faster. People got killed with those low-grade gliomas in 2-3 years. But they were anecdotal evidence. Nobody really knew why. And so when we finished the classification, we found this group that was very clearly different from the others, and I call them GBMs in training. Okay, they are glioblastomas, latent glioblastomas. And so if you can identify patients that have a latent glioblastoma, then you can have a different type of therapy for them, which prolongs their life. Not curing yet, but prolonging their life. And the classifiers needed in order to determine who were these latent glioblastomas were already FDA approved. And so that got translated to the clinic immediately. In a matter of 3-4 months after the publication in the New England Journal of Medicine, the American Medical Association and the Neuro-Oncology Society came up with guidelines saying you need to test people in low-grade gliomas with these markers, and if they have these results, you need to be a lot more vigilant and more aggressive in therapy. And so that was an immediate change. Another thing that was found in stomach cancer, there's one subtype of gastric cancer that is produced by an Epstein-Barr virus, an active Epstein-Barr virus infection, which obviously has treatment implications. And so that also was adopted very, very fast because EBV testing is FDA approved and can be applied to the clinic very, very fast. So there have been some things that have translated very, very fast. There are some hints of other stuff that will require confirmation in clinical trial samples in order for them to actually move to the clinics. But for example, one breast tumor type, it doesn't really look like breast at all. It looks like ovarian tumors. They're very, very similar, and they should be treated similarly. So that is also moving to the clinical practice.
Are drug companies accessing this? Are they?
Drug companies are accessing the data, and they are looking at the data. They have been looking at the data all the time. One of the things that happens when we give informed consents, so patients can give us the samples, one of the things that we insist on having there is that if you withdraw your consent, we will not put any new data in the database. But the data that has been already put in the database cannot be called back. The genie is out of the bottle. And the reason the genie is out of the bottle is because our database gets trolled by computer bots every single night, 200-300 of them that copy everything new that we have. And we know that about 50 of those come from pharma. So pharma is getting the data and it's looking at it. And it's good because pharma is the only entity that can really develop the drugs. They cannot do the basic research because basic research requires national funding, but they are very good at producing the drugs. And without the drugs, we cannot treat the patients, so it's a partnership.
Let's go to the questions from the audience. We have some really excellent ones, and I think you may have just answered the first one with your pharma bots trolling through the data. Is all the data now publicly available?
Yes, all the data has been publicly available. One of the things, I told you about scientific mindsets that has changed. The TCGA also has changed societal mindsets. One of the things that used to happen is you generate your data in your lab, and then you keep it in your own computer, and you look at it at night, covering it so nobody can peep onto it, which is absurd because most research is paid for by public funding, so the data should be available. When TCGA was put together, we decided that that was not going to be the model. And so we have been putting the data out in the public as soon as we know that it's technically correct. Basically, we do the QC, we make sure that the data is correct, and then it's out. So we get updates all the time, and it's always publicly available. There's 2 tiers of data. There's the freely publicly available, which is the vast majority of the data. And then there is a tier that is called control access. And the reason we have the control access is because this is data that could be used to identify the donor. And so because of HIPAA concerns, we have to keep this on a control tier. That doesn't mean that people cannot access. People can access it, but before they access it, they have to promise 2 things. They're not going to try to identify anybody, and they are not going to propagate the data outside the control sphere. And so as long as that happens, they have access to the data, and it's totally and fully available.
We had a lot of questions about CRISPR and the ability to edit genomes really inexpensively. What is the impact of something like CRISPR on this data and on the project?
God only knows. CRISPR is a very useful tool.
Does everyone here know what CRISPR is?
I'm gonna go on a little
good.
background
That's
of
good. Thank you.
what CRISPR is. So CRISPR, in very simple terms, CRISPR-Cas allows you to put a volume dial on a gene. It allows you to amplify or reduce the signal of that particular gene, which is very useful if you're studying the function of the gene, because totally obliterating the gene normally doesn't tell you much. The gene is not there, so maybe we'll tell you what it is. And having partial reductions and having a titrating reduction, basically reducing as much in steps, helps you figure out what the function is. The way we used to do this was with knockout mice. You had conditional knockout. Basically, you have a mouse that had the gene flanked by some sequences in which you could make the gene go away and basically suddenly the mouse does not have that gene. But still, it's total erasure, so it doesn't allow you to modulate. There were some modulated systems, but they were leaky, they were not that good. CRISPR-Cas is extremely efficient. And so you can say, okay, let's start with this cell with gene A at 100%, then let's go to 75, let's go to 60, let's go to 50, and then you go, oh, between 65 and 75, there was a change. Okay, so let's go from 75 to 70 to 65 to 60. You actually can do this. So it's an extremely useful tool to figure out functions in the lab. It has other applications, which is you can supplant the version of a gene by a new one. Because when you are doing the modulation, that's exactly what you're doing. You're supplanting a version from another. And so say your family is not the tallest one in the planet. There's statistics that show that people that are taller have better chances of getting jobs that are higher paying. This is true, actually, which is totally absurd, but it's the case. And so you may decide that you want to give your progeny a better chance of better jobs, and so how about if in utero we change the way the human growth hormone is modulated and give you a version that is a little more effective, and so your kids will be taller. And from there, slippery slope to very scary ideas. And so that's the issue. The issue is the tool is great. It has a lot of potential for good, but it has a lot of potential for misuse. And so, as always, what happens is science goes ahead of societal ethics. And so at some point, scientists have to stop, turn around and go, no, we're not going to do this. And so that happened with human cloning. Human cloning is possible. It's absolutely banned, but it's not banned only by national entities. It's banned by the scientific communities. There was one person that tried to do human cloning. He got shunned from the community. He got no funding whatsoever. He got not accepted. His publications was not accepted in any journal. Basically, the community said, "We told you no."
Yeah.
And no is no. And so the good thing is,
it's a kind of self-policing.
It's a
function
self-policing,
so far.
but it has the potential. It's always good to have laws in place that regulate this. And so, if somebody breaks the law, other than not having your paper published, you go to jail. That tends to be a little higher incentive.
So I gather from what you're saying, in the cancer community, especially as it relates to genomics, CRISPR is not really being used or seen as a tool that's
not
positive.
Being used for the moment as a tool in therapy. It has the potential for. The problem is it has not been studied enough to know what the side effects may be. And so that's why we're studying it. It has potential, and I don't think it will be a problem using it in a therapeutic manner, as it's not a problem to use, say, a virus to give a good copy of the CTFR gene to children with cystic fibrosis.
Yeah.
It's perfectly fine. Another thing will be using that virus as a bioweapon. Okay, so it's a question of knowing how the uses get regulated.
We had some questions about education. I want to ask a couple of those. What's the best resource for someone who's working in computer science or AI to learn about cancer if they wanted to pursue this?
I will say go to the NCI website. If you go to the NCI website, you'll find
the National Cancer Institute.
National Cancer Institute, thank you. It's basically www.nci.gov. We have funding information, but there's a resources tab that has a lot of explanations on, very basic stuff. What's a cell? What's DNA? What's RNA? How cancer appears? And then you basically have a tumor biology course there in the website. There's a lot of resources of the kind. I would start there. And then I will find a cancer researcher that has the stamina and the will to talk your ears off. I offer myself as a volunteer. And start the conversation, because that's the way we learn. As I told you earlier, when we were not in front of the audience, when we put Rembrandt together, I had no experience with computer science. So they introduced me to a bunch of computer scientists, and I said, well, we have all this data. I want you to do a database so people can do the analysis. And so they look at me and they go, what do you mean by do the analysis? Do the analysis. And they go, yeah, but you need to specify. What is that you want to look at? And I go, oh, you want me to tell you how? And that was an epiphany. And I go, okay, fine. So a week later, after the weekend, I bring them 30 questions, and they go, well, this is the good beginning, but we actually need more. And I go, oh, you actually want my questions? And so 2 weeks later, I show up with 700 and some questions. And then we started having educational sessions. I will go and talk to the computer scientist and explain what cancer biology was. And then the computer scientist will come to my lab and explain to the people that were generating the data what a computer can do and a computer cannot do. And so it was very useful because we started understanding each other. And so from that experience 10 years ago, I am a convinced person that, if somebody wants to know what you're doing, you should talk to them. Because it's not just because they're insane. Well, it may be because they're insane. But really, they want to know for a reason. And the more people we have in different fields of science that understand other fields of science,
sure,
the better we get. Yep. In fact, we have at NCI, we have a program that is the Physical Sciences Oncology program. So we are talking with physicists. Talk about rare people, okay? We're talking to physicists, and they are coming with this crazy idea of how the models could go. Things are coming out of there that are incredible. So I am a convert to talk to anybody that wants to know about cancer.
That's neat. The second question I really love, because you mentioned just as we were walking out here that you regularly talk to high school students. Mm-hmm. So here's a question that's just perfect. For a high schooler trying to choose between pure computer science, AI versus biotech, how do you see the future? Is biotech going to be a computational problem more than a biology problem?
I would say biotech is still going to be a biology problem, but with more and more input from the computer side. And whenever I give these talks at the National Library of Medicine to high schoolers about science, my message tends to be, please choose the version of scientific field most basic that you can stomach, okay? So I'm not saying that everybody needs to be a mathematician, pure mathematician, God forbid. But the more basic science education you can get, the better it is. I'm a chemist. I'm convinced that I have been able to jump from protein chemistry to cancer genetics to genomics to computer genomics and all of that stuff, is because my training is in very basic science. And so I have learned how to pull apart problems to the basic components. The more specialized you become, it's more difficult to wiggle out to a new field. And so my recommendation will be go with the most basic version of science that you can stomach. Again, if chemistry is not your thing, by all means, don't do it. As I was telling you, my oldest son, he wants to be a paleobiologist.
Right.
He's going for biology for college because paleobiology is not undergrad. And he said, well, you guys are chemists and I'm going to be a biologist. And it's like, that's what you like. Go for it, okay? But try to go for the less specialized thing. And ideally, what I would say to that person is there are bioinformatics programs at the universities that are extremely good. They used to be that bioinformaticians were really computer scientists that had taken a couple of biology classes. That's not a bioinformatician. You actually need to know biology. But now the bioinformatics programs at the universities are really well put together, and they are churning. The newer generation that I see coming to the analysis groups, I have to explain almost nothing.
I'm going to ask two final questions. The last one is going to come from the audience, but the second one isn't. The first one isn't. All this data is freely available. So here in Silicon Valley, people like to set up companies or maybe a center and start to innovate around this data because there's so many insoluble problems potentially coming out of it. Is there any approach here where just pure innovation and just getting in and mucking around in the data is actually going to produce a good outcome that you can see?
I think so. In fact, the interesting thing with the data is I see this data and I always think cancer. First question I got when I sat in the chair was, what happens to the non-human sequences? And I go, ooh. And I go, well, we don't toss anything, so there must be somewhere. So I go and ask the person that was dealing with that, and he goes, bin zero. And I go, you're going to have to explain because I don't know what bin zero means. We basically bin the sequences according to chromosomes. So 1, 2, 3, 4, 5, and then the things that are not human, they end up in bin zero, which is kind of our grandma's attic chest. We just, whatever we don't know what to put, we put it there. And so I tell the person, it's like, yeah, it's in all the BAMs, it's in bin zero. And I ask, why? And they go, oh, because you guys are sequencing the bejesus out of these samples, and we are pretty sure that there's a lot of bacteria and fungi and viruses there lurking that we could discover. And I go, oh, right. I didn't think about that. In fact, it's one of the most commonly used outside cancer use cases. The other thing that happens is we get this data because of the size of the data used for tool development. Tool development in predictions in economy. Fine, I don't know if it's the appropriate data, but it's a large data set. So using this data can be used for a lot more things than cancer. And also, as I told you, there's a lot of problems that we don't have the algorithms ready to help us solve it. And so the more people are looking at it, and this is why we make the data publicly available. We don't believe that we are the only intelligent people in the planet. We know that we are pretty dumb. And so there's a lot of intelligent people that look at it from different angles and are discovering things that we didn't even think about. And so I'll say, if there's any startup around here that wants to look at the data, go for it.
Go for it.
Yeah.
And then the last question, which a lot of people asked, if you look, knowing what you know as a chemist, as a now an expert in genomics and cancer and the mechanism for doing all of this, if you look down the road 5, 10, 15 years from now, what do you hope will happen as the result of this project and the things that are now coming after it?
Well, one of the things that I get this question a lot, so
sure.
I have an answer for it. I think that by the time I retire, I'm 50, okay? So I have about 20 years to go, or 17. I think by the time I retire, there will be a lot of tumor types that will not be a problem anymore. I'm not going to say all cancer. One question that I get all the time is, when are we going to get the cure for cancer? And my answer is never. because cancer is not one disease, it's about 300 different diseases. So you are going to have 300 different cures. And some of them will be cures. We have certain tumor types that today we cured thanks to genomics. Diffuse large B-cell lymphoma, most common lymphoma there is, used to be a death sentence. Nowadays, 95% of people with diffuse large B-cell lymphoma get cured at the community clinic level, not even specialized cancer center level, because we can classify it correctly and we can treat it correctly. And so this is going to happen for some diseases. And some other diseases, unfortunately, because they are very difficult to treat because the cells acquire mutations that make them very sneaky, they're going to become a chronic disease. So we will have certain type of cancers that we're going to have to be vigilant. We're going to have therapies. We'll start with one line of therapy. When that doesn't work, we'll go to the next one, and so on, and so on, and so on. Like we treat, say, diabetes. Okay? We don't cure diabetes, but people live long lives with diabetes. And so I think we're going to get there. And I think this is just the first step in that direction. But I am fairly confident that if humanity survives, in 20 years, we are going to see a lot less suffering from cancer.
That's great. Thank you so much, JC, for being here with
pleasure.
us tonight. You can watch again on Facebook. You can send this to your friends and have them watch it on Facebook. And I hope you'll be here for more of our programs in the weeks and months to come. Thanks, everyone. Thank you very much.
My pleasure.